I think for clinicians, what they should know is the availability of a lot of really exciting drugs. And the real question these days is, how do we position them? You know, do we want to put alkylators after BTK inhibitors, for instance, because we don't want to induce TP53 alterations? I think that's important as we discuss really the pros and cons of BTK inhibitors. There's also a lot of excitement in how we can optimize BTK inhibitor usage. How can we create a finite duration of therapy as opposed to an indefinite duration of therapy with BTK inhibitors? Unfortunately, one of the important trials, the combination of ibrutinib and venetoclax, was terminated early because of a high incidence of ventricular arrhythmias. But on the other hand, we saw a very exciting study that was presented at ICML this past summer from Neil Berenstein that taught us that we can combine BTK inhibitors, in this case acalabrutinib, along with bendamustine and rituximab. And this combination actually produces, a lot of VGPRs, even CRs, and may actually give us the opportunity to come up with a finite duration of therapy. And there's also a lot of excitement around the use of ibrutinib and bendamustine rituximab, as well as zanubrutinib and benda-R. Those are trials that are in some stage of being launched at this point, and we'll be excited to see what those combinations produce. I think what I'm also keeping an eye on is how we can use CXCR4 antagonists, because we clearly have a CXCR4 problem. 40% of patients carry an activating CXCR4 mutation. And we know from the phase 1 study that I ran that was reported already in blood that you can use an antagonist to CXCR4 and combine it with a BTK inhibitor and actually see the results that are very much in line with somebody who didn't have a CXCR4 mutation. So I think being able to, specifically personalize drugs based on the mutational profile can be achieved. And this is why I'm excited. That's what we're keeping an eye on.